This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV, This Week in Virology, episode 1265, recorded on October 24th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Oh, I forgot to pull up the weather, but it's kind of gray, and the weather is 50 degrees. Fahrenheit feels like 42, and I can't do that Fahrenheit to conversion that quickly, but let's see what it tells me here on a different app. Oh, spinning. 9 degrees feels like 7 Celsius. So
here in New York, it's 15 C and cloudy, feels like 11. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. I'm not sure what it is right now. It's probably what my weather app tells me, which is 88 degrees, which is cool.
Cool, right.
And it's going to rain tonight, they say.
Oh.
They're all liars, but that's what they say. That would be really welcome. I haven't seen Skywater for weeks, so we're looking forward to that.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And here in Western Mass, it's overcast. We just had some sprinkles of rain roll through. But otherwise, it's been lovely fall weather, 53 Fahrenheit, 12 C, and we are right around peak foliage right now. Beautiful.
Peak foliage. I was in Virginia last week, and they're nowhere near peak foliage.
Oh, no. We've just, our maples are beautiful right now. They're red and gold, and the oaks are just starting to turn, so they're mostly green, but they're going to be brown soon.
You enjoy these programs about science. We're in an era where everyone needs to know more science. We would love your support, because that's what we do here. We give you science. We go right to the original papers and talk about them. And we'd love your support to continue this work. You can go to microbe.tv/contribute. No news today, except I just want to remind everyone that we will have Immune 100 live at the incubator on December 22nd at 6 p.m. And if you go to microbe.tv website, you'll see on the front page a link where you can buy tickets. So some of you may remember, we did TWiV 1000 at a theater here in New York. You had to buy tickets for that. They're a donation. But after that, I swore I would never do any of those things again. And that's why we're doing this at the incubator.
TWiV 1000 was like planning a wedding almost. It was,
Well, I did it myself. I didn't have anyone help me, because there was no one here at the incubator at the time. And then we had a lot of technical problems when we got there. Although it ended up working, and it was great. But I didn't want to do it again. But now I have two people here, Kim and Valerie, who are helping out, and they're taking care of everything, like the publicity. All the Eventbrite thing was done by Kim. We have to rent chairs, 25 chairs. Valerie's going to take care of that. So that's great. I can do it here at the incubator. And so everybody will be there. We have 2 guests from New York City, so it should be fun. And there are different levels of tickets, too. You get different gifts for how much money you spend. We're going to give you stuff to take home.
How often does Immune happen? Trying to figure out how many years 100 episodes is.
Yeah, it's basically once a month.
Okay, so we're talking 8 years?
Yeah, it's taken a long time to get to 100, yeah. It was like 5 years for TWiV. Remember, we did 100 in Johnson City, Texas. And you were there, actually.
Yeah, that's right.
But
that was the last time I saw Neva.
Yes, yeah, she was there, right.
She was there, and I had no clue. She was gone within months or weeks from that.
Yep. Well, the only reason TWiV reached 1,000 so quickly was because of COVID.
Because
We
of
started
the
doing three
pandemic.
a week. We were doing 3 a week at some
3
point.
a
When
week.
Daniel split
Yes.
off,
Yeah.
and we would do 2 of these episodes, and then Daniel would do his. Oh my gosh! And we really accelerated because I remember at the beginning of the pandemic we were at 600, and then all of a sudden, this is April 2023. We did 1,000. Oh my gosh! I was thinking back the other day about the early days of the pandemic. Oh my God, you forget so much that happened. It's
I
amazing.
listened to a very early episode of TWiV recently. I forget what it was or why, but it's great. Good stuff. Long history.
Well, yeah, we had that first episode in January where we were wondering what was going to happen, right? It was good. This little virus went to market.
Yep.
We have 2 very, very, very interesting papers today, which, sometimes the work we do is not obviously directly relevant to every man, but these are, I think. And the first one, I had originally made it a snippet, but it's not really a snippet.
It's a big snippet.
It's a big snippet. I said, well, this is cancer. We'll snippetize it because we don't really understand all of it, but it's actually really good.
Well,
It's
cancer, you usually want a surgeon to snippet it.
It's right. This is a Nature article. SARS-CoV-2 mRNA vaccines sensitize tumors to immune checkpoint blockade. Anybody want to do the authors?
Yeah, so the authors, there are 4 co-first authors, Adam Grippen, Cristiano Marconi, Sage Kopling, and Nan Lee. 2 senior authors, Elias Sayur and Stephen Lin. And a list of affiliations appears at the end of the paper. So I scroll. Damn it, Nature, why do you do this? Oh, yes. Now, here we get to the affiliations. They're from University of Texas MD Anderson Cancer Center in Houston, University of Florida, Gainesville, McGovern Medical School in Houston, Baylor College of Medicine in Houston, Stanford University, Scripps Health in San Diego, and University of North Carolina, Chapel Hill.
So, Kathy, you want to give us a brief summary?
Yes. So the title may have been the briefest summary, but I think I have a better way to say it. And that is basically they're finding that the SARS-CoV-2 mRNA vaccination can improve immune therapy that's used for some cancers. So the original thing that kind of tipped them off to this, maybe at least they kind of alluded to this in the introduction, is that there have been case reports of patients whose tumors spontaneously resolved after COVID-19 mRNA vaccines. So what they did in this paper was evaluate a bunch of existing data on people with either small cell lung cancer or melanoma, 2 different kinds of cancer. And they found that indeed, people who received mRNA vaccines within 100 days of an immune checkpoint inhibitor therapy lived about twice as long on average as people who got the immune checkpoint inhibitor therapy, but no vaccine. And they didn't see that for non-mRNA vaccines, so for flu vaccines or things like that. So the finding is that if somebody's getting this immune checkpoint inhibitor therapy and within 100 days they got a mRNA vaccine, which we'll find out doesn't have to be a SARS-CoV-2 one, then if they got that, then they had a much better survival, about twice as long. So they then expanded this study to mice, and as I just alluded to, they found it wasn't just specific to the spike in the SARS-CoV-2 mRNA vaccine, but they found it with another antigen in an mRNA vaccine. And they found that this seems to be because these vaccines are inducing chemokines and cytokines, particularly interferon alpha, and those rev up the body's immune system that then stimulates CD8 T cells, the killer T cells, that can then target tumor antigens. So that's the mechanism. And so a person has to be getting this immune checkpoint inhibitor therapy for maximal efficacy. So since this is TWiV and not immune, and because I wasn't really even sure what this immune checkpoint inhibitor therapy was, I want to explain that a little bit. So cancer cells produce what are called checkpoint proteins. And one thing I found said you can think of them as shields. So the cancer cells produce these shields to basically thwart the immune response. And if you treat somebody with a checkpoint inhibitor, it breaks down the shield or removes the shield, or you can even think of it as removes the brake on the immune system being active. And so this is one of the breakthroughs in cancer therapy of recent years. And so this immune checkpoint is getting inhibited by these therapies. And in about at least 50% of the cases, it doesn't work because there's a sluggish response by the patient's immune system, even when this brake is taken off or the shield is broken or whatever. And so they would really like to remedy that. And so one way to do that is what are called personalized cancer vaccines. So they identify specific targets on a person's cancer cells and try to design a therapy based on that. But that's laborious and expensive, as you might imagine. So in this paper, the idea is that maybe this just general mRNA vaccine that stimulates chemokines and cytokines can rev up the sluggish immune system and make this immune checkpoint inhibitor therapy work better. So my final summary, a part of this is once again just to say that this SARS-CoV-2 or other antigen mRNA vaccination can improve this immune therapy that's used at least for some cancers.
Yeah. So the personalized therapy that you referred to, that would be like CAR T-cell therapy, right?
Right.
Right. So the analogy I would use here is it's the accelerator and the brake. So the T cell, if you've got a response going on against the tumor, and the immune system is active against it, the tumor generates these checkpoint proteins. It's like it's slamming the brakes on the T cells, and then you block that checkpoint. You take the foot off the brake, and now the T cells can kill the tumor. However, if you don't already have a robust immune response, or you're not mounting a robust immune response against the tumor when you start this therapy, taking your foot off the brake, you're just going to kind of coast forward in neutral because the foot's not on the gas.
Yeah.
Whereas here, it seems what we're going to see is that you can put your foot on the gas in many ways.
So I want to check my thinking here on immune checkpoint inhibitors, because I'm not sure if I'm making this up or not. The cancer cells didn't just make up this immune checkpoint stuff out of thin air. It's like your immune system gets ramped up in terms of making more cells and doing all kinds of stuff when it encounters a challenge, right? But then ultimately it's got to tamp that down. It's got to turn off. Otherwise, you've got leukemia or something all the time with your immune cells going bonkers. So the checkpoint proteins or mechanisms are a normal part of the immune response.
Sure.
And in tumor cells, I would guess probably have a microevolutionary advantage that they're taking of this by making their own inhibitors to thwart the immune system trying to get them. Fair enough?
Yeah.
Right.
Yes.
Okay.
Right. So the tumor creates an immunosuppressive microenvironment in itself,
Right.
But does that with natural components that the immune system would normally use to
Right.
blunt the response.
It's most likely one of the dozen or so mutations that you need to make a cancer cell has to do with checkpoint inhibition. Yeah.
I go way back to when I did a postdoc at the Imperial Cancer Research Fund.
Mm-hmm.
And there was always an ongoing conversation about immune surveillance and the notion that you're constantly getting cancer and your immune system is on guard, looking out for this. and squashing them. And yeah, I could never quite fully believe this, but certainly these sorts of observations with tumor cells and the immune therapies speak to that whole notion.
Kathy, what were you going to say?
Oh, just that when you started to explain it, Rich, that the cells just don't make this checkpoint protein so that cancer cells can take advantage of it. It reminds me of saying cells don't make receptors just for viruses.
Exactly.
You know?
Exactly.
So just to get a little more granular, there are a couple of different kinds of checkpoint proteins. One of them is PD1, which is on the T cell, and PDL1, which is on the tumor cell. And if they engage, then the T cell doesn't kill the tumor cell. And so you can make antibodies against either PDL1 or PD1, and that's the immune therapy. You give people these antibodies. There's also another one called CTLA4. So you're giving them antibodies. And they work with certain kinds of tumors, but then again, they don't work in all patients, as you already heard, right? And the goal is to try and improve it, because as they say here, we don't have any methods to improve these immunotherapy approaches that modify the tumor microenvironment. But there was a clue with the vaccines, the mRNA vaccines, and they're trying to pursue that. And this is another example of a paper. We did a paper on TWiV last week, which combined epidemiological with studies in mice. And this does that as well. We have an epidemiological study which suggests something. It's an observational study which doesn't prove anything, but it makes suggestions. And then they go into a mouse model and the suggestions are proven. And then they go back into humans and try and validate some aspects of it. All right, so they have a cohort of patients who have stage 3, 4 non-small cell lung cancer. They're being treated at the MD Anderson Cancer Center. This is January 2015 to September 2022. They identified 180 patients who got a COVID mRNA vaccine within 100 days of starting their immune checkpoint inhibition therapy. All right.
And I do want to call out the love of abbreviations in this paper. I mean, yeah, you sometimes want to abbreviate stuff, but does tumor microenvironments really need to be called TMEs and immune checkpoint, ICI, and overall survival is OS?
That they didn't explain.
Right.
And I had to just glean what it meant.
Yeah. Yes. And then they even mentioned their institution, the MD Anderson Cancer Center, MDACC. Guys, stop. Just, you don't need all these abbreviations.
Well, and as I pointed out to Vincent before we started, in the figure 1 where they use this overall survival and progression-free survival, they abbreviate those. They have room on the graph to have spelled them out.
Yes.
And it would have been so much more informative for the reader who just wants to look at the figures.
Yes.
You know.
Probably the whole manuscript was, there's so many authors, they're all over the place. They wrote the manuscript by text message.
That's right. Anyway, so 180 patients got a vaccine with 100 days of initiation of immunotherapy. That's the key here. And 704 who were treated with immunotherapy but didn't get a COVID vaccine. And they had people who got different numbers of vaccines. They got the Pfizer or the Moderna vaccine. They got 1 dose or they got 2 doses. It's 3 doses, etc. But basically, they all got some kind of mRNA vaccine or not. And then they control for 39 covariables, right? This is the latest thing now. I think this was also in our paper that we did on how the VZV vaccine decreases dementia a couple of weeks ago.
Yeah.
Lots of covariables like clinical stage histology, steroid use, performance status, mutation status, comorbidities, treatment year. They basically find that if you got a COVID mRNA vaccine within 100 days of starting your immune therapy, it's associated with a improved median overall survival, 20 months versus 37 months. And then the 3-year overall survival, 30.8 versus 55.7%. So it's a good effect, right? 20 versus 37 months. And this survival advantage was similar for stage 3 and stage 4 patients, right? They stage tumors. The higher the number, the more screwed you are. And so 4 is really bad. But so 4, so stage 3, unresectable. So you can't have surgery. It's not going to help you. And even then, you saw a survival advantage with the mRNA vaccines. And 2 vaccines in 100 days, similar extension of life. I can't even remember what OS is, overall survival.
Overall survival.
Gosh, some vaccines you hardly use, some abbreviations, you hardly use them, so you never get to know what they are. So 1 or 2 vaccines got the same result. Patients who got a vaccine within 100 days of chemotherapy. So this is the group of patients who weren't candidates for immunotherapy, had no survival benefit. So the vaccine alone is not cutting it, right?
So this seems to be specific to immune checkpoint therapy.
Yes. And if you got it's also specific to mRNA vaccines, because
Yes.
if you got a pneumonia or flu vaccine within the 100 days, didn't have any improvement in survival.
Oh, and by the way, we were talking about pneumonia vaccines last time or time before. I have since learned that the recommended age for the pneumococcal vaccine is now 50.
Okay.
I learned that at my doctor's office when he said, "You're due for this." I'm not that old. He said, "Yes, you are." So, got it.
They repeated the whole analysis in patients who had metastatic melanoma. And again, you have an improved overall survival, 26 months versus 36 months. Both Pfizer and Moderna work, and 1 or 2 vaccine doses work as well. And you're going to see later that there's a difference in the manufacturer, and I want to talk a little bit about that because hopefully I'll remember to do it. Okay, so what's the mechanism? So now they go into animals to try and figure out the mechanism, because you can't do that in people. And so they made their own COVID mRNA
I thought this
vaccines.
was cool.
Yeah.
I mean, they had to go through a lot to make their own.
Yeah.
I guess it's for legal reasons, right? They can't just buy vaccine. But you're giving it to mice. What difference would it make, right? I don't know.
I don't know
why they
I don't know either.
So anyway. So
maybe you can't just call up Moderna or Pfizer, or maybe both companies didn't want to give them vaccine for mice, or didn't have extra supply, or
sure they would have bought it.
Whatever.
But anyway, so they have mouse models for melanoma and lung cancer, right? And then so they treat and these mice do not respond well to immunotherapy, right? If you give them checkpoint inhibitors, they don't respond well. So they treat them with 2 vaccine doses together with the immune checkpoint, the immune therapy. And it really helps. It's superior to either monotherapy alone in terms of restricting tumor growth. So they measure the tumor size, right? And that's restricted. And also, if you have established tumors in mice, they metastasize, and the metastases are inhibited by the immunization as well. So the result extends to this mouse model, which is cool. Okay, so now you have a mouse model where you can ask, where you know that the mRNA vaccine improves tumor resolution. So what's the mechanism? So they say maybe cytokines are involved. There's some evidence that cytokines might be involved, and so they try cytokine blocking antibodies. They try antibodies against the IL-1 and interferon alpha receptor. So IL-1 receptor blockade had no effect. But if they use antibodies to the interferon alpha receptor, it completely blocks the positive effect of the vaccine. And if they give the mice a lot of interferon, it recapitulates the anti-tumor effects of the vaccine. So somehow interferon is involved in the mechanism of action of the mRNA vaccines on immune therapy for cancer. So what about other mRNAs? So they have an mRNA vaccine encoding a cytomegalovirus antigen. They get the same effect. It also works, right? So it doesn't matter what's encoded in the mRNA. It's the mRNA itself. Although it would have been interesting if they had put a stop code on somewhere or took away the start code on in the mRNA vaccine and saw what happened. But that's really too basic science for these guys.
Or just some random RNA polymer.
Yeah.
It's bad enough they already had to make one vaccine.
Yeah, I understand. Anyway, so as you know, the COVID mRNA vaccines have N-methylpseudouracil in place of uridine, right? To reduce innate immune activation, right? Because that way the mRNA vaccines are around long enough to give you an immune response. So they replaced the N-methylpseudouridine with uridine in their vaccines, and you get even more synergy with the immunotherapy, which makes sense because now if it's interferons involved, you should be getting more interferon responses.
Maybe they anticipated wanting to do that experiment in advance, and that's why they made their own vaccine.
Maybe that's right.
Because if you used off-the-shelf vaccine initially and then you used this unmodified, you wouldn't really be able to compare the two.
Yeah.
But they really only saw that with the CMV cytomegalovirus one. They didn't see it with the...
Yeah, that's interesting. That is really interesting because...
With the spike.
So they say the specific mRNA must make a difference also, yeah. Okay, so how does this work? They wondered if somehow it was double-stranded RNA in the preparation. So they go through great lengths to get rid of all the double-stranded RNA in their preparations, and it still has anti-tumor efficacy. And if they take mice who lack a gene encoding a protein called RIG-I, which is a sensor of double-stranded RNA. So the RIG-I will sense double-stranded RNA and activate interferon production. There's no difference in the efficacy of the vaccine in promoting tumor resolution. So it's not double-stranded RNA that's mediating this effect. They also took the same RNA and made lipid particle aggregates instead of lipid nanoparticles, right? Anionic lipoplexes, lipid particle aggregates. And they did the same assay, and they don't work. They don't have anti-tumor effect. And so that what they said, they extracted the RNA from their vaccine, lipid nanoparticles, and they find that the RNA is present in high molecular weight secondary structures, similar in mass to double-stranded RNA, although it's not. And so they wonder if it's complexed with protein or something, and that's what's actually stimulating the production of type 1 interferon. So obviously you could do a lot more there, but they stop at that point. Everybody good so far?
Yeah.
Yeah. So it's and maybe we haven't gotten here yet. It's not just the RNA. It's not just the lipid nanoparticle. It's the RNA in a lipid nanoparticle.
Yes.
And I kept thinking the whole time, one of the differences, and we'll get to this, between Pfizer and Moderna is the actual composition of the lipid nanoparticle.
That's right.
Also, there's a dosage difference as
That's
well.
right.
We want to keep that stuff in mind.
Yeah,
And I don't know what the composition of their lipid nanoparticle was. I don't know that they can. I don't know what the, whether they know or can know what the composition of the Pfizer and Moderna lipid nanoparticles are. So there's some fuzzy there, but importantly, it's the lipid nanoparticle and the RNA together that's important. Yeah. I mean, this got me. I was describing this to some friends of mine the other day, and I was reminded of the idea that the coolest reaction in science is not eureka. It's, oh, that's interesting.
It's interesting. That's right.
That's funny.
This is one of these. This is like serendipity, right?
Yeah.
Just falling out of the sky.
Okay, so let's go more deeper into mechanisms. So they find that when you treat the mice with RNA, LNP, lipid nanoparticles, you get interferon alpha elevated and other T helper chemokines. So there's a surge in cytokine chemokine response. And they also see activation of myeloid cells in lymphoid organs of mice. They see activation of antigen-presenting cells like dendritic cells, macrophages, and also a subset of antigen-presenting cells called LY6C plus cells. They have MHC class 2 on their surface. And if you blockade the interferon receptor, all of these activations or these increases are abrogated. And so you can't just add interferon. It doesn't do the same thing as a lipid nanoparticle, okay? So interferon, it's more than just interferon. And this myeloid cell activation is extended to the tumor, not just systemically, but the tumor itself, where the lipid nanoparticles can go, and they are associated with interferon alpha receptor dependent increases in these LY6C positive myeloid cells. And so they wonder if these are cells that are getting enhanced presentation of tumor antigens via major histocompatibility proteins, MHC1. So they actually study presentation of ovalbumin, a test protein, a test antigen on MHC1. And they find that when you give mice the spike-encoding RNA, lipid nanoparticles, they stimulate antigen-presenting cells to present tumor antigens in lymphoid organs in the presence of co-stimulatory molecules, which is the way presentation normally occurs. And this is partially enriched in these LY6C plus MHC2 positive cells. So a little bit about the mechanism of what is going on there. Okay, so lots of activation of antigen-presenting cells, expansion of CD8 positive T cells as well, and their activation as well. And along with that, PD-1 expression, so remember, PD-1 is on the T cell, and PD-L1 is on the tumor cell. So
PD-1 is a receptor, right, for PD-L1?
Yeah, it is,
Yes.
but they're both membrane proteins, right? But yeah, PD-1 is the receptor for PD-L1, so PD-1 is on the tumor cell. Okay, I lost my paper. Here we go.
Program
So the
death
tumor
1 and
cell
program
uses...
death ligand 1.
Excuse me?
Yeah,
It's program death 1 and program death ligand 1.
right. Yeah, PD-1 goes up in T cells. And so this therapy is priming T cells. So there's the summary of all this is there's not just local tumor immunomodulation, but systemic immunomodulation, and that these systemic cells go to the tumor. Or these myeloid cells go to not just the tumor, the lymphoid organs to present tumor antigen to T cells, and you get activation of T cells, and they can go to the tumor and work on it as well. Okay, so they want to show that the spike lipid nanoparticles, spike RNA lipid nanoparticles was responsible for expanding tumor-reactive T cells. Remember, so what I've just told you is that tumor-reactive T cells are now expanding. They isolate CD8 positive T cells from the spleens of the mice that have been treated, and they stain them with what's called a tetramer, which is basically 4 MHC molecules with peptides, which if they're recognized by T cells, they will stain the T cell. So if the T cell's got a T cell receptor for a specific peptide, the tetramer will stain it. And so basically the combination therapy with the checkpoint, with the immune, what's the C stand for again?
Immune checkpoint inhibitor?
Yeah.
No, immune, is that it?
Yeah.
Immune checkpoint inhibitor, ICI.
Okay, the immune checkpoint inhibitor.
ICI, Vincent.
Yes, right. Just call it ICI.
I see eye to eye. I see eye to eye with you. The immune combination therapy with the ICI and let's just say ICI to hell with it. And the lipid nanoparticles stimulates the expansion of T cells as determined by tetramer staining for each of these antigens that are melanoma-associated antigens they looked for here, right? Tumor-specific antigens. And they used another assay to show an increase in, again, activation of CD8-positive T cells. So CD8 T cells are the cytotoxic T cells that would kill the tumor normally, and they increase after the combination therapy. So it's mediated by interferon, but the end result is that you're making lots of new CTLs, cytotoxic T cells, that can go and kill the tumor, and also because you're adding the ICI, which is going to block the interaction
checkpoint
of
inhibition.
Yep. Checkpoint inhibition. Okay. So then they looked at the T cells more specifically in these treated mice, and you can see an increase in tumor infiltrating CD8 positive T cells in the LNP ICI group greater than 20-fold. A lot more T cells going into these tumors, and they're reactive with these tumor antigens that they've looked for. Now here, originally I got confused here, but I shouldn't have been if I had read the introduction properly. So they say, commensurate with the increase in tumor-reactive T cells, we found that spike encoding vaccines, right, significantly increased PD-L1 expression on tumor cells. And blockade of interferon alpha signaling blocked that PD-L1 expression. So that's important for the immunotherapy response. So in other words, spike RNA LNP stimulate the production and infiltration of activated T cells, tumor-reactive, tumor-specific T cells that overcome the compensatory expression of PD-1 and PD-L1 in the presence of checkpoint inhibitors. So you're making a lot of T cells that are overcoming the PD-1, PD-L1 interaction. So I was confused by increased PD-L1 expression on tumor cells. I'm like, but you don't want PD.
Yeah, exactly.
L1. But we decided that that's how checkpoint inhibition works, right?
Yes. So the increased, my understanding is that the increased PD-L1 on the tumor cells is indicating that you're actually getting the immune response that you want. You're standing on the accelerator.
Right.
So the immune response, the T cells are coming in there. The tumor then has to turn on PD-L1 to stop them, to say nothing to see here. And then that is what sets you up for the checkpoint inhibition therapy, which is going to...
That's the key. You need to then checkpoint the PD-L1
right.
and PD...
PD-L1 has to be there for you to use this.
But
so if those pathways aren't happening, you're wasting your time.
Yes.
Okay.
Yeah. Yes. So that's why you have to have a certain percentage of
PD-L1 positive.
PD-L1 in the tumor for you even to get immune checkpoint, ICI.
Okay.
Otherwise, it's not going to work. And then here, you're turning up the amount, so with your nanoparticle vaccine. And as you'll see in a moment, that can help a lot.
Yeah.
So, okay, so all this is in mice. So now they say, as there are differences between humans and mice, actually, they say differences in how humans and mice respond to mRNA, which is really true. We did a paper on that a long time ago. So they want to see if similar things are happening in humans. So they get blood and plasma from 5 people at baseline in 6 24/7 days and 14 days after getting the Moderna vaccine.
These are healthy volunteers. As far as we know, they don't have cancer.
Yeah, as far as we know. So and they saw that interferon alpha was upregulated in them. Most upregulated cytokine at any time point. It increased 280-fold to baseline. And also IL-6 and interferon gamma were elevated as well. And a bunch of inflammatory cytokines surged at 24 hours after immunization. So next time you get a vaccine, you can think of all these things: interferon alpha, gamma, epsilon, CXCL10.
When I get a COVID-19 vaccine, I already had a pretty good idea that the cytokines were going nuts afterwards.
So when
Now you know exactly
Now
which
I know
ones.
which ones.
when I get one, I always say, "Go ribosomes, go!" And then
But
now I should say, "Okay, cytokines, chemokines, come in."
all this is finished in 7 days. They returned to baseline at 7 days. So if you're getting all those, fever, lethargy, whatever after
It'll go away.
it should go away in a week.
Mine actually resolves within like 24 hours usually.
Yeah. Okay, then they wanted to know the effect of this vaccination on immune cells. So they looked at circulating myeloid cells by flow cytometry, and they found that the mRNA vaccine drives innate immune activation because you get increased PD-L1 on circulating myeloid cells and dendritic cells. They also see activation of natural killer cells and circulating T cells by their flow cytometry. So basically, and finally, they have some volunteers who got Comirnaty, the Pfizer vaccine, and they looked at them, and they found similar changes to the cytokine profile, but the magnitude of increase was significantly less with the Pfizer vaccine because it has less mRNA compared to the Moderna vaccine. And I'm thinking the latest Moderna has even less mRNA in it. So it's not going to be as good for this tumor effect.
But you could modify that for cancer treatment.
Well, I think they're going to make specific
Yeah,
vaccines.
sure.
They're not going to use COVID
No,
vaccines
no.
for this, right? They're going to optimize it. But you need to have a certain amount of RNA in there.
Well, I think at least anecdotally, if not, I don't know to what extent this has been measured, the response to the Moderna vaccine in terms of side effects was more dramatic than the Pfizer. That's what it makes me think of. So, if a lot of people in particular reacted very strongly to the Moderna vaccine and did better with the Pfizer vaccine.
Okay, now this is cool here. So they say that the ability of T cells to get into tumors depends on PD-L1 expression. All right. So they made this. They hypothesized that patients who get the COVID vaccine, they should have higher PD-L1 expression on their tumors, right? So they had first they have pathology reports from 2,315 patients who had non-small cell lung cancer who had biopsies. And when you do the biopsy, you look for a number of things. One of them is the tumor proportion score. What percent of the cells have PD-L1 on the tumor? It's L1, right? On
L1,
the tumor,
yeah.
On the tumor. So you can quantify that because they look with an antibody for presence of the protein. Patients who got the COVID vaccine less than 100 days before their biopsy had a 24% increase in PD-L1 compared to patients who had not gotten any vaccine. 24% increase. So it's actually increasing the PD-L1, which is that's part of the mechanism. It's very cool. And the reason this is important, I didn't know this because I'm a virologist. This tumor proportion score, which is how much PD-L1 is on the tumor related to the cells, 50% is the threshold to determine whether patients are going to get immunotherapy versus chemotherapy. And so they found that patients who had gotten a COVID-19 vaccine were 29% more likely to meet or exceed the 50% threshold over unvaccinated patients. So in other words, having an mRNA vaccine will impact on the kind of treatment you get. All right. Because it's based on the amount of PD-L1. And if you had a influenza or pneumonia vaccine, it makes no difference on this tumor proportion score. Is that what? Yeah, tumor proportion score. So I thought that was very cool.
Yeah.
Okay, so I'm still, I have new confusion about PD-L1.
It's okay. It's okay.
You said something that hadn't sunk in for me, that in order to gain access to the tumor, the T cells that are expressing PD-1 actually need to grab onto PD-L1 in the tumor?
So the phrase they used is infiltration of T cells into tumors is associated with increases in PD-L1.
Associated with increases in PD-L1.
The T cells don't need it, but when the T cells go in, the tumors are more likely to express PD-L1 to defend themselves.
That makes more sense.
Yeah.
Because
that's my reading of it.
I still have this thing in my mind that PD-L1 is actually an inhibitor.
Yes, it is.
Of the T cells, right?
Yes,
but
it is.
It also is a marker.
Yes.
For what's going on. Okay. All right, good.
Yeah, because it's probably induced by the T cell, right? Or is it always up there? It's not always up there, is it? Or are tumors in a normal environment? It's probably regulated, but a tumor is probably just on there. Well, it's not, because it's only in a certain fraction of people do that.
The tumor, you select for tumors that are going to express this when the T cells go in, because they're going to be able to take them out otherwise.
Yeah, but baseline, because that's why this tumor proportion score is an important indicator for therapy, because if it's too low, they're not going to bother because the checkpoint therapy won't work, right? I'm still not clear about if it's turned on by the T cells or not.
That I don't know. I don't know if this is a specific response the cells mount when they encounter the T cells PD-1, or if this is something that the cells
but the observation is for the T cells getting into the tumor is associated with PD-L1, and it says correlated with anti-tumor response in preclinical models, which would be an animal model. Okay.
I'm calling for a lifeline here. You listeners out there in TWiV land, we need mail. Okay.
Yeah, if you're some oncologists out there who understand this, sure, write in. This is a bunch of virologists, fumbling with oncology, but it's really interesting, and I love it.
Occasionally, we get mail on issues like this that clarifies the whole thing.
But we are able to understand this at a reasonable level.
Yeah.
There are always facts that we don't know.
We have our limits.
We're also able to say, huh?
Yeah.
I don't understand that.
So a letter from an oncologist who understands this would be great.
Okay, so now they're going to go beyond non-small cell lung cancer and melanoma. So they have a cohort of patients who have come into these cancer hospitals, and they have pathology reports, and they search them for the term PD-L1 for all the pathology reports. In a 4-year period, including the pandemic, they identify 5,317 pathology reports from January 2020 to October 2023.
These are presumably at MDACC.
Yeah, MDACC with the term PD-L1. And a diverse variety of patients with a variety of primary sites and histologies. Receipt in this cohort, receipt of a COVID vaccine within 100 days before biopsy was associated with a 37% increase in tumor proportion score. That is, the fraction of tumors that are producing PD-L1. Okay, and they didn't see this increase with influenza vaccines. So then they repeat this. At another cohort, in their cohort, for survival, and they found patients who received any COVID vaccine within 100 days of initiating this ICI experienced improved survival relative to the unvaccinated peers. Okay, so in people, some aspects of what they found in mice also is in people as well. Okay, and then there's this term which I found interesting, a cold tumor.
Yeah.
Immunologically cold, right? So there's not much of an immune response in the tumor. So they want to know if vaccination could restore immune sensitivity to these immunologically cold tumors. So they use pre-vaccine TPS as a surrogate for immune sensitivity because, again, people with TPS of less than 1% have reduced benefit from
That's the
ICI,
tumor proportion score.
right, compared to people with a baseline above 1%. And so among patients with stage 4 non-small cell lung cancer and baseline TPS less than 1%, those who got a COVID vaccine within 100 days of getting ICI exhibited an overall survival similar to that of patients with a baseline greater than 1%, suggesting that the vaccination somehow restores sensitivity to the immunotherapy.
Warming up the cold tumor.
Yes.
It's warming up the cold tumor. Right. So that's That's very cool. So basically, here let's summarize it. mRNA vaccines, and the antigens they encode are not tumor antigens, right? They're just spike in this case. You get stimulation of anti-tumor immune responses. They sensitize tumors to ICIs. And so how they do that, the mRNA vaccines make a surge in antiviral chemokines. It drives innate immune activation. Then tumor-resident innate immune cells are activated by these cytokines. They prime T cells. They become activated. They go into the tumor, and the tumors are making PD-L1 to evade the T cell attack, but the immune checkpoint inhibition neutralizes that. So together with the COVID vaccines, you get tumor regression and improved survival. That's cool.
Yeah,
that's
very cool.
But I have to say, warming up cold tumors sounds like the worst possible meal of leftovers.
So, first of all, this has been in the news.
Yeah.
Mm-hmm.
A lot of listeners will have seen headlines on this, and I just want to point out that if you've seen the news briefs on this, this is the paper they're talking about.
Yeah.
Okay.
And this paper is open access.
And secondly, this is one of these things where this is just the beginning.
Yeah.
This is an observation that you can hopefully tweak all over the place to improve cancer therapies.
Well,
Very cool.
they identified some tweaks that you could do just in this paper. I mean, you can boost it in a couple of ways just from here. I mean, this is a tremendous finding in the context of checkpoint inhibition therapy, because what people have been working on are, as Vincent said at the beginning, these custom tailored approaches, which are going to be, I mean, the cost of that is yodelingly high, where you've got to take each patient and customize a therapy in the lab and then come back to the patient. It'll never be available to anybody but the richest, and that's just, it's cumbersome. Whereas here, there's this finding that there's a potential to have a one-size-fits-all vaccine. Okay, you're coming in for checkpoint therapy in a month. Here's your vaccine, and then go.
It's an interesting statement they make at the end. These results position off-the-shelf RNA therapeutics targeting infectious disease antigens as universal modulators of anti-tumor immunity. I'm just wondering what the mRNA will look like when they finally license this for people, right? I mean, the COVID vaccines work. Maybe you could make it better, but...
well, in fact, they kind of allude to, we've got all this really cheaply made, high volume vaccine. Maybe we should just use that. That seemed to be one of their positive points about it.
I wonder if you could get better results though with different.
Probably.
That'll be a line of research.
Yeah.
And they do at least imply in this that there may be a structural component in the RNA, or in the RNA as packaged in the lipid nanoparticle that makes a difference. So it could be that structural component is somehow sequence dependent. So who knows? I wouldn't be surprised if there was some optimal RNA structure. I wouldn't even say sequence, but structure that helps with this, and that'll determine the sequences that are used down the road.
So basically, you could develop a rapid assay, because the key here is induction of cytokines, right?
Mm-hmm.
So you could do it in mice, but you make a whole pile of different RNAs and throw them in and see which one works. I don't know how you'd figure out which one it is.
There's probably even some library approach, right, where you start with a mix,
that's what I'm thinking.
Right?
Some library approach, because you want to get the best induction, right?
Yeah.
Even
before we get to those optimizations, there are ways that this could be optimized, and I assume people are going to. But right now, as somebody who's been diagnosed with cancer, and the immune checkpoint therapy might be a possibility, I am not making medical advice, but if that happened to me, I might run out to the pharmacy and get a COVID-19 shot because it's available.
Yeah.
Yeah, if you were going to get ICI, right?
Yeah,
sure. I mean, you could say to your oncologist, what do you think?
Yeah. Oh, definitely. Yeah.
And they could
talk
say,
to an actual doctor about this.
They could say, well, it's not licensed, but there's no reason why you can't go get a COVID vaccine.
Nope.
In fact, we have them right here. We could give it to you.
Yeah.
for sure. Anyway, I thought it was a very cool paper.
Yep.
Very, very
Great.
cool.
Yeah.
I think the results are very clear too.
Yeah.
Right. Okay, so this one is pretty much a pure virology paper, but I'm still confused about certain things. But that's life. This is another Nature article. A vaccine central in AH5 influenza antigenic space confers broad immunity. So we're going to take a trip to the solar system.
Take a trip to antigenic space and find a central point in antigenic space. So the first author is Adinda Koch, and the senior author is Matilda Richard. These folks conveniently listed at the bottom of the first page, Nature, thank you very much, I see you can do it sometimes, are from Erasmus University Medical Center in Rotterdam, University of Cambridge in UK, Utrecht University, Netherlands, University of Melbourne, Australia, King's College London, and University of Wisconsin-Madison.
And we have two people who have been on TWiV here. Derek Smith, who is one of the founders of antigenic cartography, which we'll talk about, and Ron Fouché, who has worked on influenza viruses for years. So, Kathy, a little summary?
Sure. So the top line summary, they're going to use this antigenic mapping technique to define influenza antigens that confer broad immunity, and they're specifically looking at the avian influenza hemagglutinin H or HA. So the problem is that antigenic evolution of the H5 avian influenza viruses is difficult to understand and predict because the underlying drivers or factors are unclear. Now, in humans, population immunity buildup helps with understanding evolutionary patterns of flu viruses, but this doesn't occur in animals. For instance, they point out there's no real herd immunity buildup due to the low likelihood of reinfection because of segregation of populations, shorter lifespans, virus lethality for the birds, and rapid renewal of susceptible populations. So, in the absence of being able to understand this in the same kind of way that you can do it for humans, and because understanding these avian influenza H5 viruses is important, they use an approach that's not based on viral genetics, but is based on virus antigenicity. So they create this antigenic map using a lot of existing data, and then they define and test some possible antigens that they can identify from this mapping approach. And they try to see whether they can generate broad immunity against a variety of avian H5 hemagglutinins. And they succeed in this, and they use immunization and virus challenge in ferrets as their animal model.
So as Kathy said, the H5 avian influenza viruses have been undergoing antigenic drift, just like others. And I didn't know this, but the WHO biannually, what is that, once every 2 years?
I can never keep
Twice a year.
Twice a year.
Biennial is once every 2 years.
Biennial. Okay. Biannually, twice a year.
Twice a year.
Select H5 vaccine candidates. And so since 2006, 48 candidates have been recommended. Just in case it became a pandemic, you would have a candidate.
You'd have a recommendation
vaccine,
ready to go.
Right.
The H5 influenza viruses that are now global, spread all over the place, and are changing all the time, are a prime candidate for a pandemic virus. So we need to be on the lookout on these. This is important work.
Right.
No, these are the ones that are in cattle that we keep hearing about, and they're everywhere.
Yep.
They say, this is an interesting thing, while continuous updating of these, another abbreviation, candidate vaccine viruses, CVVs. And while it's necessary, they say it's a reactive and unsustainable approach to, prepare for pandemic preparedness. We need, because vaccine preparation takes 6 months, and these vaccines that are currently made by inactivation are low immunogenicity. So, because humans are immunologically naive against avian influenza viruses, so they say we need something better. And so that's what they're going to try and do here. They're going to look at H5 hemagglutinin antigenic evolution, which they say has never been done. But that's the kind of thing we need to make a high-resolution antigenic map, use a big collection of viruses, and see if we can make select antigens that might induce a broad neutralizing response. So what they do is they have 127 HA genes of subclades along the phylogenetic tree of H5 influenza viruses. All right. And that's for one of the figures is the phylogenetic tree. You can see it's a very broad swath of everything that's out there. And then they make ferret antisera against 33 of these viruses. They pick 33 of them that are diverged genetically and antigenically. And so they end up with 127 antigens and 33 ferret sera against these 33 viruses. And they cross-titrate them in hemagglutination inhibition assay. All right, so they have the antibodies and the viruses, and they do hemagglutination inhibition assays, not infectivity yet, to see how the sera are cross-reacting. And then they create what's called an antigenic map or antigenic cartography.
Yes. And the reason they're doing this instead of just comparing sequences is because you don't really care so much what the sequence differences are. You really care what the immune response to those changes is. And so they actually need to map the antigens, and that's how they do that. What cross-reacts with what.
And that's what makes this study different
Yes.
than so many others, is you're looking actually at the reactivity, which is kind of duh.
But it's also
I don't mean that as knocking them. I'm thinking, that's what you need to do.
That's what you need to do, but it's a heck of a lot of work.
Yeah, right.
And they did.
In fact, what they have here, they have a three-dimensional antigenic map. So previously, the antigenic map would be the HI titer, the hemagglutination inhibition titer, so the antibody can inhibit hemagglutination inhibition versus the virus, right? So the particular virus isolate versus the HI titer. So here they have a three-dimensional representation. And each direction, so X, Y, Z now, we have a three-dimensional map. It represents antigenic difference. And one square of this grid corresponds to one antigenic unit, which is a twofold change in HI titer.
It's actually one cube of this grid since it's three-dimensional, but yes.
It's one cube, yes.
And in the supplementary data,
they say square, though. Yeah,
you can get these figures that you can twist around in space so that you can see the three dimensions.
And I will now admit that I neglected to read as deeply because I got to the supplementary data. I said, I'll check that out. And I spent the next, I don't know how long, just, like twisting this thing around. Ooh, cool.
So the distances in the antigenic maps are inversely correlated with HI titers. So antigens close in space with sera against them, they react with an high H titer, and further away have less reactivity. And you can also see from the map the antigenic relatedness between the antigens. That's not directly measured in the HI assay, right? So that's another dimension, the antigenic relatedness. And that's what was said earlier. You don't care about the sequence of the RNA. You care about the antigenic relatedness of them. Okay, so they make this three-dimensional map, and they find that they remove some of the antigens because they're not cross-reactive enough. And the final map has 117 antigen and 29 sera on it. And so then they let's see here. They used a subset of these. They didn't want to use all of them to do neutralization assays. And they find that the neutralization assay results correlate with the HI titers. Okay, so that's good. And that's the video now we could look at.
And the one thing that pops out, especially in the video, which adds these antigens in their spatial map on the antigen map over time.
Right.
And the point there is that the map is non-directional, as they put it in the paper. So you don't see an evolution of the virus from one area of the antigenic map to another area of the antigenic map over time. You see it moving across and over here and back here and just kind of ping-ponging around in this space, which is critical for what they're going to do next.
One of the things they found, which is interesting, is a discordance between genotype and antigenic phenotype. So if they say, for example, antigens from different genetic subclades are sometimes really close in antigenic space. So that's why the antigenic cartography is so useful. Now, the other thing they find with this is that the maximum distance between we neglected to say that the avian H5N1 influenza viruses originated in 1996 in Guangdong. So those are called GSGD antigens. And then there's some avian influenza viruses which are not originating there. They're called non-GSGD. So they say the maximum pairwise distance between the non-GSGD HAs, 43 years, is only 5.58 antigen units, but the GSGD antigens spanning 25 years have 14, the maximum pairwise distance, 14.7 antigen units. So there's an increased level of antigenic evolution relative to time in the GSGD lineage compared with that of non-GSGD. So let me explain better. So the GSGD lineage started in 1996, where it first arose in Hong Kong and Guangdong and spread globally. And then you have non-GSGD viruses that have also been spreading on their own. Okay. So the GSGD is really antigenically distinct. And so they say that's...
I'm thinking that it's creating faster a greater diversity of antigenic specificities, right? And my guess is that is why it has been so successful spreading globally.
Well,
does that make sense?
Compared to non-GSGD,
right.
As we'll see later, actually, overall in the influenza space, it's less diversity because of the unique problems and properties of birds. We'll talk about that.
Right. But the GSGD is, as Christopher Walken would say in that SNL skit, it's really exploring the antigenic space.
It is. And that's why it's a challenge to design a vaccine that could be protective of any of these antigens. Right.
But it's exploring that space in a non-directional fashion, so it bounces around.
So they say that what we can do maybe is to take some antigens that are at the center of this space, and maybe they would induce antibodies that would neutralize many others. And so they say that's the purpose of the antigenic map that we have made. They say theoretically, high cross-reactivity could be achieved in a non-directional space with a single antigen, provided that it elicits an immune response centered in antigenic space, and it's highly immunogenic. So they say the maximum distance of antigens from the center of the map has not increased substantially since 2010, even though the virus is continuing to evolve. So they say maybe the H5 space is constrained, and so maybe that would help us. So we're gonna check that out. So what they did, they looked for antigens within 2.5 antigenic units of the center, and the center is the mean of all the antigens that they have there, right? So they got 12 HA antigens there, which react with more sera at a higher titer than those further away from the center. So if you take those 12 and sera against them, they will react with more sera than ones further away. And 11 of those, interestingly, 11 of these 12 lack a glycosylation site at amino acid 154, and that is present in 37% of all the antigens on this map. This has been known to increase vaccine immunogenicity. And some of so that's one thing to keep in mind, that glycosylation site. The other thing is that there are amino acid changes that are consistent with expanding the receptor binding ability of the HA. So avian HAs typically bind to alpha-2,3 linked sialic acids, but there's some amino acid changes in these that are consistent with binding alpha-2,6 amino acids. And they say, well, if it's going to be a pandemic, it's got to bind alpha-2,6. And so we should put those amino acids into our candidate antigen, whatever it may be. So they end up picking a virus HA, Anhui 2005. It's a virus isolated in 2005. And they introduced these amino acid changes to make it bind both 2,3 and 2,6. And they test that and a couple of other antigens against all the sera in their data set. They vaccinate and they decide that these are good candidates, these central antigens. And then they vaccinate and boost ferrets with vaccines comprising these HAs. And they have a whole bunch of controls. And then they take the sera from the ferrets and they titrate against all of their antigens. And basically this Anhui that they have modified to bind alpha-2,6 sialic acids, and I think also to have to lack that glycosylation site. This antibodies against that react with the most antigens in their data set. They pick that, and they're going to say, let's check this out in ferrets and see how it works. Okay? So now we have a central antigen, Anhui, which has been further modified by introducing some more amino acids. And antibodies against it react with more of the antigens in the map than anything else. So they're going to check this out in ferrets. So the goal is to look at how it protects ferrets compared with what they call standard of care, which would be the homologous virus, right? And the HI antibody response and protection against challenge. And there's a lot of controls here. all kinds of viruses. They have different clades. First of all, they have 2.2.1.2, and they have 2.3.4.4a, which I believe is the one in dairy cattle.
Maybe,
yeah. And they even have an H5. And they chose these viruses because the HAs are distinct from one another. They're about 10 antigenic units apart from the non-central Anhui virus. And they vaccinate ferrets twice, 28 days apart. They have 6 ferrets. And then they use the two vaccines, which are the standard of care. That's they're homologous to the challenge viruses. So the challenge viruses are called A. duck Giza 2015 and A. Sichuan 2014. Okay. And they have different neuraminidase so that you rule out any participation of the neuraminidase, because we know neuraminidase antibodies can be important as well. So you rule out any participation in that. Okay. And so they adjuvanted these vaccines because they say that they're not going to be very immunogenic in humans, so they have to have an adjuvant. So they boost, and then they get serum, and they measure the HI titer. And all the animals make HI antibodies, and they titrate their sera against the 113 antigens of their antigenic map. And this AC Anhui vaccine, that serum was positioned closer to the center of the map than the one raised with the non-central wild-type antigen. So some of them were non-central, and those don't work as well as the central one in making broadly reactive antibodies. And they introduced changes that they put into Anhui, the glycosylation in the receptor binding, led to a more central and enhanced immune response as well. It's very interesting. Interestingly, the parent virus, Anhui vaccine, did not make detectable HI antibodies against the 2 challenge viruses, the Giza and the Sichuan challenge viruses. But we'll see that still animals were protected in a bit. But the AC is the modified Anhui. It induced HI titers in all the ferrets, and had a comparable response with the homologous challenge, the homologous vaccines. Then after boosting, the ferrets are inoculated. They're challenged now intranasally with Giza or Sichuan virus, right? So now you have ferrets that have been vaccinated with either Sichuan or Giza and Anhui or Anhui AC, the modified Anhui. They look at symptoms, they collect nose and throat swabs, they measure the body weight. So the mock vaccinated ferrets who didn't get any vaccine, they have a big body weight loss. They have reduced activity. Some of them die. In fact, they have virus in their lungs. They're shedding virus. But vaccinated animals have lower pathological signs. They have lower weight loss. They don't look as badly as the others. They have lower virus levels in the lower respiratory tract. So the vaccination protects against movement of the virus from the upper tract to the lower tract. So, and the vaccination of all of these viruses reduces disease severity and replication in the lower tract for all the vaccines. There are some differences between the three vaccines they used, but basically the point is that this Anhui is non-inferior to the homologous vaccines against viruses of two very different clades, right? You got this clade and this clade that are far apart. And if you homologously vaccinate against them, you get the same result as if you do the Anhui vaccine. So that means it's got an enhanced broadness of protection.
Right.
So I have a question because this non-inferior caught my eye several times. And there's a reason, I guess, why they're avoiding just saying superior. Non-inferior.
Or similar. It's not superior necessarily.
Could be equal to or better. Yeah. Okay.
It could be equal to or better. Yeah.
Right.
Not worse. If it's worse, then they haven't done what they wanted to do.
Right. So that non-inferior is a term from drug development that's like the term you would use in this case, because you don't want to say they're the same because they're not exactly the same, but they're within the statistical margin where you would say it's not worse to get
Yeah, it's non-inferior.
So they're being conservative in what they're saying.
So essentially, that's the proof of concept here, that you could take a central antigen and get protection against two different non-central antigens from different clades, validating this antigenic map approach.
Yeah, because normally if you vaccinate with a non-homologous vaccine, and you see this in the Giza versus Sichuan, you vaccinate with Giza, challenge with Sichuan, and that is inferior because you used a different vaccine.
Yeah.
But if you vaccinate with Anhui and challenge with either of these, you get a response that, yeah, Kathy, I would say it's equivalent, but they can't technically say it's equivalent. It's non-inferior.
What's interesting here is so these ferrets were immunized intramuscularly, and with one of the combinations, let's see, which one was this? Infectious virus. In the mock group, the mean increase in body temperature was higher compared with that of Anhui and Giza vaccine groups. Infectious virus was recovered from nose and throat swabs of ferrets from all groups, corroborating that intramuscular immunization does not prevent upper respiratory tract infection. I just wanted to mention that because some of the vaccines protected against disease, but they don't protect against infection, which we always say about vaccines. But this is a nice experimental. And the ferrets were challenged at 28 days, right? Very soon after vaccination. So the point here is that you can make an antigenic map and of many, many different H5 HAs and pick one that will give you broad protection. And so they say this is just a proof of concept, right?
Yeah.
And but as H5 continues to evolve, we should do this periodically and have say this is our candidate for this 6 months, right? And then move on to the next one.
Or maybe even say this is our candidate for the next, till we do this again in another year.
Yeah.
Instead of biannually, you could maybe do this less often. And they also make the point that if you actually made some of this vaccine and had it sitting around, you could, well, they don't say it this way, but the idea is you could roll out this vaccine faster because you already know the antigen,
right.
And then respond with a specific vaccine if you needed to, but you'd have that initial, you could have one vaccine that would give you a good response initially.
So it's a way of using this cartography to evaluate. So what you want is a antigenically central antigen, right? An H5 antigen, which is what they picked here. But they also say, we don't have to just use inactivated vaccines like we did here. We did that because that's what are used for influenza virus. But you could use other technologies to make antigens and
mRNA.
Different vaccine platforms, mRNAs or other kinds of vaccines, vector-based vaccines. They say you should still research that, but at the same time, use this cartography as a way to pick the HA. And by the way, they say we should do the same thing with NA, because we know that increasing evidence indicates that NA antibodies are really important. And so why don't we do antigenic cartography for NA and see which one is the central NA and could be incorporated as well. So this is the first time this has been done, right? Yeah. Previously, people take sera and isolates and they do neutralization assays and HIs and they decide the best match. But this is kind of a more systematic way of getting at the right antigen.
So check me on this one too. This another thing I've been thinking is we've done a number of different papers that identify by various routes broadly neutralizing antibodies.
Right.
And a question that comes up in my mind that I think I vocalized on this show before was, this is great, but how do you make an antigen that will do that? And to me, this paper is the answer to that. This is how you make the antigen that gets you the broadly neutralizing antibodies.
Yeah. Now, the bad news here, if you're reading this paper and you're saying, oh, great, why don't we do this for seasonal flu? The answer is because seasonal flu, the antigenic map shows directional movement because it's under selection every year, and the virus adapts to that and moves away from the space it was in. And so if you did something that was, back in the middle, you would miss probably the current virus. Doesn't mean that it's a bad idea to map the antigenic space of where the seasonal flus are, but what they kind of get out in the discussion is that they seem to be implying that this may not work as well there.
So I forgot to mention the animal discussion they had here, which is, so in humans, as Kathy said, we have immunity and that selects for antigenic variants. But it's different in animals because the animals don't live as long, right? So if they get immune, they're going to be killed. So they're not going to be in the pool to do selection, right? Yeah. And so...
but especially if you're talking about poultry farms.
Poultry farms in particular.
You don't have the same chicken around next year to get the flu again.
But now it's established in wild bird species, many wild bird species. So that may now give us different patterns of antigenic evolution in the future, right?
But they still imply that there's the spatial and, temporal distance that is going to keep that from happening, it sounded like.
Yeah, we just don't know, right?
Yeah.
They also say that antigenic evolution of these H5s could be partially a bystander effect of other adaptive processes, like adaptation to systemically replicate or to host species switching, which is interesting, right? The idea that you go to a new species and you select for a change that's needed for efficient replication in that species, and that also would give you an antigenic change, which is not unreasonable, right? It's very interesting. I like it very much. Okay, let's see. Let's do one round of email. I think we have enough time. Alan, can you take that first one?
Okay. John writes, Doctors TWiV, you mentioned sudden death while walking to work with that solar panel that blew off the roof. I don't know if you know the sad story behind the major contributing factor in Roger Hendrick's death. His wife was off to a summer music camp at Oberlin with their only car. and it was a nice day, so he walked 1.5 miles to the lab. Crossing an intersection with the light not far from their home, the light changed. The car at the intersection waited, but the guy behind him, somehow not seeing Roger and in a fit of impatience, whipped around that car and hit Roger. He was in severe shock and nearly didn't survive, but he managed to return to the lab not quite the same. He died a few years later, and all are convinced that the accident was the primary contributing factor. The tangential reference to orders of magnitude triggered the memory that my Swedish postdoctoral mentor, Hans Jernvall, the characters didn't quite come through there, but Jernvall is the pronunciation, was fond of saying that in his lifetime, the sensitivity of Edman protein sequencing had improved by 9 orders of magnitude, and that record would never be beaten, since there were only 7 left before getting to single molecules. Okay. Re the CDC firings, how would Group Orange even recognize a mistake? I also recall the letter you received back in about February from an anonymous in the Kansas City office, where EIS officers who had been tracing a TB outbreak were suddenly not able to travel. I wonder how TB's doing there. When kids predictably start dying from vaccine-preventable diseases this winter, the response should be, so what? It's just kids. However many. To the inevitable aghast reply, just note that's their response to school shootings. And I randomly came across this regarding the actress Jean Turney. Early in her stardom, she was pregnant and at a party in 1943 when a fan came up to her and said that she had broken quarantine for German measles to come meet her. Turney contracted rubella from the fan, and her daughter was born deaf and with other defects, which played a heavy mental toll on Jean. I've condensed this post that appeared in my Facebook stream. In May 2024, residents of Douglas County, Colorado, were stunned to see a mountain lion staggering across a property, its hind legs barely working as it dragged its body along the ground. Wildlife officials rushed to the scene, but the big cat's condition was so poor that Colorado Parks and Wildlife chose to euthanize the cat. Tests revealed the cougar had been suffering from staggering disease, a rare and fatal neurological disorder caused by the Rustrella virus. This marks the first known case of the disease in North America, previously only recorded in European domestic cats and a few zoo animals. Digging into this a bit, I learned that Rustrella is a rubella relative. It's possible that it has been mentioned on TWiV, but I don't recall, and came up empty on a pointed search. What's the level of concern for Rustrella? Best wishes from a rather chilly Greater Braddock, where a low of 2C is expected this Saturday. Thank you, John. I don't think we've talked about Rustrella.
I just looked it up, and it's the cause of staggering disease.
Staggering disease.
Right, that's a new one.
Yeah.
Never heard of that. I'm not aware that it's a zoonotic concern. I don't know.
For those who may not be aware, Roger Hendricks was a phage scientist of significant repute. I think he has a lot to do with, what's it called? The phage hunters program, et cetera.
Oh, C-phages.
Yeah.
Yeah, I didn't know that this is what happened to him. He was on TWiM many years ago. He studied the evolution of phage in a little pool of water in a rock near his house, near the lab.
When I think of Roger, I think of the phage. They make up weird names for their phages, and one of the originals was corn dog, because the thing actually looked like a corn dog.
That's right. All right, so Kathy, can you take? Maybe you could take 2.
Okay. David writes, Hi, in case you missed it, the protein design folks are hosting a contest to design proteins that neutralize Nipah virus. I'd be interested in your thoughts on the assessments that they are planning. And he gives the link. And David is from Ann Arbor, and I think that might be David States we've heard from before.
It is.
Yeah. Okay.
It is.
So, you can...
They didn't say... So in the post, there's an X post. They don't really say what they're going to test. They say design proteins capable of neutralizing Nipah. We will synthesize and test 1,000 of the most promising proteins. I mean, I guess you could design proteins to bind the glycoproteins on the surface, right?
Yeah, if you click on that thing, there's instructions on how to do it. But like you say,
There's no assessment, so I don't know what assessments they're planning other than neutralization, right? But it's an interesting. 1,000 candidates is not bad. It's cool.
Do you want me to do the next one?
Yeah, please.
Mary writes, Dear TWiV team, thank you for all you do. Who knew there was a World Food Prize? It was founded by a Nobel laureate who won the Nobel for his work on worldwide hunger. I'm delighted to find out about this prize and to share the story of this brave woman agronomist who overcame early opposition to pursue her career to eventually bring a bacteria to bear on solving human hunger. Her accomplishment improves food crop yields and lessens the need for expensive manufactured fertilizers. Well done. And Mary provides the link, and Mary's a regular listener.
Cool. Rich.
Let's see. Hang on. Peter writes, greetings. Jolene mentioned Ribosome Studio for biochem animations. The Clockwork Show also does some great animated explainers. Recently, this one on allergens, and she gives a link. The paper discussed would be a good topic for a more in-depth MicrobeTV podcast. Fun stuff. Thanks, Peter.
I think you should take the next one too, Rich.
I got hung up in the video there. Okay.
Yeah.
Somebody else did too. So, okay. Zach writes, hello, TWiV. Greetings from sunny Brisbane, Australia, where it's going to get up to 29 C later this afternoon, and where I'm a medical student with an interest in infectious diseases. I just got finished listening to TWiV 1263 and was interested in the idea that we should have a smallpox eradication day. There is one.
Is there?
Well, no. It's Smallpox Vaccination Day. It commemorates the day that Jenner vaccinated, what was his name? James Phipps.
Yeah.
Okay, 200 years ago.
What day is it?
I have to look it up. I will look it up while the next email is being read. Okay. Because I've got it on my calendar.
Sorry. Continue.
Sadly, I think one reason we're unlikely to see that is that an honest appraisal of that effort would require us to honor the largely unsung efforts of scientists and diplomats from the USSR. Whatever your thoughts are on the evil empire, the eradication program itself was first proposed by Soviet representatives at the WHA, and the WHO website today says they provided hundreds of millions, all told more than 1.55 billion, freeze-dried vaccines that were the basis of eradication in China, India, and Eastern Europe. Considering that these are the most populated places on Earth and that global population in 1979 was only just passing 4 billion, it's impossible to imagine this effort succeeding without the Soviets' contribution of vaccines for nearly half the world on their own. Everyone knows about Sabin and Salk, and even Salk's quip about the patenting of his vaccine. Could you patent the sun? Unfortunately, I think American society is too embittered still from the Cold War to recognize the real contributions of Soviet scientists like Viktor Zhdanov and their collaboration with epidemiologists from the U.S. like Bill Fogarty to accomplish this world historic feat. It's a triumphant story for all involved, and sadly, it won't be told the way it should because we, the U.S. that is, would have to share the glory, something we have an ever-diminishing capacity for, with a vanquished foe no less. Love the show, been a listener for a long time. Best regards, Zach. I don't think we're that evil. I think we can do this, okay? And I think it's a delightful opportunity to, to be global.
Yeah.
Let's do that.
Thank you. So the contribution of the Soviet Union to the polio vaccines is well acknowledged. There are many articles about Sabin going over there and collaborating with Soviet virologist Chumakov, right? So I don't think it's a problem, yeah.
No, I don't think that's a limitation.
And one from John. As if there weren't enough outages already, Harvard cut science PhD admissions by 75% for the next 2 years. This is an article in the Harvard Crimson. So it's across all programs. And I heard about this when I went to the Harvard Virology retreat. And they said, yeah, it has to be cut because of financial reasons. But they told us it could be 0 or 75%. Which would you want? It could be cut 100% or 75%. I mean, it's better than 0, right? Having 0. But yeah, they'll have to take fewer students, which is unfortunate.
That sucks.
Because science is what drives everything. Yeah, and it's true. You can argue all you want, and you're wrong. Science is really at the base of everything that societies do.
So smallpox vaccination day is May 14th.
May 14th.
I don't know that this is official, but it was May 14th in 1796 that Edward Jenner inoculated 8-year-old James Phipps with material from a cowpox lesion. But I didn't put this into my calendar, okay? Apple did it, or somebody. So it shows up for me every year, May 14th.
And someone who wrote the letter suggested, I think, December 10th, because that's the date that WHO declared eradication.
Yeah, I've always had trouble with
December 9th.
Yeah, I think you have to do the WHO declaration. There are several dates out there, like the last case of smallpox and et cetera. So I get confused, and some of them are a couple of years apart. But I think the WHO declaration would be an appropriate date.
Yeah. And make it official. Make it an actual
Make it official.
proclamation on the calendar.
Yep.
I don't know how you make a day. I have no idea.
I think you have to lobby Congress. Good
Yeah,
luck
you
with
lobby Congress, and people in other countries lobby their governments. But this is something that can be done.
Not with this Congress.
Well, no, I mean, the government's shut down and nothing's getting done. But at some point in the future, we will have a functioning government again, I think.
It's going to be a TWiV campaign.
Yes.
We can all go to D.C. and lobby Congress for a smallpox eradication day.
Okay, then, let's do some picks of the week. Kathy, what do you have for us?
Well, this article caught my eye from the New England Journal of Medicine, which I think is a free article. Permethrin treated baby raps for malaria prevention. And I think it's a really cool story. And then it turns out that I'm not up on my TWIPs, and it was covered in TWIP 266 pretty recently. And so permethrin is a chrysanthemum extract, or a synthetic version thereof, that you can use to impregnate cloth, and it actually will kill insects and ticks, bed bugs, mosquitoes, things like that. And so the cool thing about this story is that in a double-blind, randomized, placebo-controlled trial in Uganda, they gave women with children 6 to 18 months old permethrin-treated wraps to wrap their babies in, or sham-treated wraps. And bottom line is they worked. They had mild rashes in the treated group, 8.5% versus 6% in the control. But anyway, the incidence of malaria was 0.73 cases per 100 person weeks in the treated group, and 2.14 cases in the control. So about 1/3 as many cases in the treated group. So that's really great, and was pretty feasible to do. The women and children visited the clinics every 2 weeks to get health questionnaires and so forth. And anyway, this came up in part of interest to me because I use permethrin when I'm going to go on hiking, backpacking, etc. types of trips. And you only use it on your clothing. You don't ever put it on your skin, and you treat your clothes not when you're wearing them. And Sawyer is the company that makes it. It comes in this yellow bottle. I get it at REI. And you can treat your clothes, and it will last, they say, for 6 weeks or 6 washings. It's mechanical agitation that knocks it off. And so it's a really cool thing to treat your clothes with it. And one caution they mention is don't use it near your cats. And, of course, don't pour it down the drain. But anyway, I had some reason to get more information, and I wrote to their customer service, and the person wrote back a really nice, detailed, thorough answer, which I appreciate. So that's my story about permethrin.
I could expound at length about permethrin, pyrethroids and permethrin and pyrethrum. My grandfather worked on this. He was actually
right.
Developed
Right.
some of the early versions of these.
Right. So
Big fan.
But and then the second thing that just came along for me is a movie called Phil Sharp and the Biotech Revolution, and our RNA society, whatever it's called here, at the University of Michigan, sponsored this in the local theater that plays cinema as opposed to movies, and so this. And if you went, you got a book, a hardbound book written by the writer of the screenplay, I guess, as it were. And it's interviews with Phil Sharp, and who won the Nobel Prize for with Rich Roberts for RNA splicing, but also his effects or his activities in the biotechnology space, Biogen, and a subsequent company that he helped found. There are interviews with his wife, his children, people from his lab, colleagues, and so forth. And so it really is kind of a biotech, but it's also a kind of a nice story of that series of things happening in his lifetime. And you can watch it on PBS online. And so that's why I wanted to include it. You don't just have to go to a special showing of it. And it's a pretty upbeat movie, film, cinema, whatever. I think they said it was in the can as of last November. So that was before any of the things started happening with our government and science and so forth. So there you have it. But I learned a lot of things about Phil Sharp, and that was fun.
Cool. Yeah, I watched this. It's pretty good. I always knew that Phil grew up on a farm in Kansas, but the story is Kentucky. The story is much more dramatic than that. Born in a cabin with a dirt floor type of stuff. Okay. Yeah. And so it's a great rags to riches story as well.
Mm-hmm. Cool. Rich, what do you have for us?
All right. I have a Wikipedia article about that has wider implications about a woman named Lydia Fairchild, and I will do the sneaky approach to this. So Lydia Fairchild, born in 1976, in 2002, when she was pregnant with her third child, was undergoing a separation with her husband. And according to the local sort of legal procedures, there had to be just routine paternity tests to verify that they were the parents of the children. And the paternity test came back for the two children that had been born, that the husband was indeed the father, but that she was not the mother. Okay? And she said, nonsense. But she was faced with stuff like, taking away her kids, fraud, or being a surrogate mother or something like that. And so they really honed in on this and actually had somebody present at the birth of her third child, and took appropriate samples from him and from her and got the same result. Okay, that she was not the mother, but
despite having given birth right in front of the
witness.
That's right.
Yeah, but the father was the father. Her lawyer, brilliantly, learned of a similar thing in a New England Journal of Medicine article, which I also link here, a phenomenon called tetragametic chimerization, which is a phenomenon where at conception is basically fraternal twins. You have two embryos, so it's two ova and two different sperms, so tetragametic, and that in a very early embryonic stage, these things fuse. And so you wind up with a chimeric individual where you can have whole blocks of tissue that are from one of the two twins and the rest from another twin. And in this particular case, it's the reproductive tissue that was from one twin and the whole rest of the organism, the Lydia Fairchild, from the other twin. So that when they sampled her blood or whatever other, cheek swab, whatever other tissue, they got DNA from the twin that was not the genome of her reproductive tissue. And in fact, when they went back and swabbed her cervix, they got a match for the kids. Okay?
I think they also determined that the grandmother, her mother, was the grandmother of the child.
Right. And I have not yet done the inheritance pattern for that, because I could. That was just beyond me. But I thought that I had no idea.
Wow.
That this phenomenon existed, and the story behind it, I thought was.
So she was a twin then, right?
Well, yeah.
Yeah.
She was a twin.
She still
was
is.
Two people. Still is.
Yeah. No, but she was.
No.
When she was born, was it just her or?
No, just her.
Just her.
Just
but it
her.
says
well,
here
it's
that
two of them.
This happens when you get,
You get, but the
two
embryos fuse at a very early age, at a
like
very early stage.
blastocysts.
So you no longer
So who is the other contributor to her?
The twin that got absorbed.
the twin that got absorbed.
I see.
I mean, she's a hybrid. She's a chimera. I don't think you can say. I suppose her brain. I don't know. What's her identity? It's two. Really,
Yeah.
genetically.
Most of her, you said most of her somatic tissues are.
Most of her
one thing.
somatic tissues are of one genotype, but that genotype is different than the genotype of her reproductive tissues.
Yeah.
Well, this paper, the New England Journal, is slightly different. Maybe it's a different woman.
It is a different.
Yeah.
She had renal failure and she got a
Yes.
HLA testing, and it said she wasn't the mother of her kids.
Exactly. The lawyer discovered that paper in the process of trying to defend Lydia Fairchild and figure out what the heck was going on. Good on the lawyer.
Yeah.
Good. Well, good for them for taking samples at birth also, right?
Yeah.
Yeah.
That's fascinating. Wow, I had no idea.
No
Very
idea.
cool. Very cool. Wow.
And I have
So-
no idea how I came across that. I have an email folder and a bookmark folder, both labeled TWiV pics, and stuff shows up and I just drop them in there.
I have a very similar arrangement.
Alan, what do you have for us?
This is another self-promotional one. I'm not going to do this a lot because this is actually the only other fiction story I've written so far. But this is a link to it. It's a direct link, so you can get past the paywall. So yes, indeed, I have a Nature paper. This is in the Nature Futures column. They started a few years ago doing flash fiction, which is under 1,000 words. So you can read this thing in about 5 minutes. And I hope people will. So that's a direct link. It's free, and enjoy.
I read it. I enjoy it very much.
Thank you.
Very
Me
nice.
too. Yeah.
I think the whole
Don't
theme-
spoil it.
Okay, I won't say anything.
Okay.
No, because the theme, the general theme, the genre, that particular micro genre of science fiction, I really enjoy.
Okay.
My pick is a website called Radio Garden, which may have been picked before. I don't know.
I haven't seen it. News
This
to me.
looks a lot like something that I picked years ago, but it's gotten a lot better if it's the same thing.
Well, it's basically it's a site where you have a globe and there are dots in many places. If you click on them, it plays the local radio station.
Yeah.
So you can pick a city anywhere or pick a dot and you will find a radio station in some other place.
Some radio total nerd put that together.
Yeah.
Yes. I realize that most people don't listen to radio much anymore, but you can go all over and see what's playing music, It's pretty cool.
Pretty
Thanks
cool.
to Kim, who is our social media manager, for pointing that out.
That is neat.
We have a listener pick from Jamie. Hello, Twivsters. My husband was recommended a mobile game that I thought the aviation nerds of TWiV would enjoy. It's called Sky Cards by Flight Radar 24.
Or
Flight radar.
Flight radar.
I think it's radar. I use Flight Radar 24 all the time.
And can be found in your app store. It's like playing Pokemon, but with airplanes and actual flight radar data. You can catch various flights worldwide, assemble a deck of aircraft cards that are then used to battle friends in quick matches with randomized rule categories. Fun and educational.
Good.
Okay. There you go, Alan. Check it out.
Cool.
It'll do it. For TWiV 1265, you can find the show notes at microbe.tv/TWiV. You can send us questions, comments, pics to TWiV at microbe.tv. And if you enjoy these programs, we'd love your support. Microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan, Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Conda is an Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. So this is the old TWiV crew here. We're united, right? So it was me and then Dixon and then Alan and then Rich and then Kathy in that order. So, here we are just by chance back together. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.